Good morning, one and all. I am Dr. S. Marina Prem Kumari, working as Professor Biotechnology at Agriculture College and Research Institute, Madurai. Today I am going to give a lecture on the topic Media and Culture Conditions and Sterilization Techniques. So there is no definite media for plant tissue culture. You cannot use a single media for all the explants. For different types of explants, you have to use various types of medium. So this table shows you about the various types of medium that has been developed for each and every culture. For example, it is the scientist Noop. He formulated the salt solution in 1865. Then Upensky and Upenskaya for the culture medium for algae in 1925. Gotheret, he formulated the callus culture media in 1939. And White, he formulated the root culture media for tomato roots in 1943. Murashki and Skoog medium, this is a very important medium, most commonly used, but not for all cultures. We cannot use it for all the cultures. We have to change the medium based on the explant. But mostly it is used, MS medium. This is a basic medium for tissue culture and callus culture, formulated in 1962 by Murashki and Skoog. And niche medium for anther culture in 1969. Then Gamberg for soybean culture medium 1969, and Schenck and Hildebrand medium for callus culture of monocots and dicots in 1972, and Lloyd and Markgown for woody plant medium, it is in 1980. So now we will see into the components of the plant tissue culture media. So these are the 5 important components in plant tissue culture media. One is the water. Water is very important. It needs to be demineralized or deionized water. Then next is the organic components, that is the carbon source, vitamins as well as the amino acids are used, and also some complex organic substances are also added. Next is the inorganic components, which comprises of the macronutrients, micronutrients as well as the iron source. And fourthly, it is the plant growth regulators like auxins, cytokinins, gibberellins, abscisic acid and ethylene, and gelling agents are agar, agarose and gellangrams. We will see and elaborate about each and every components. First of all, it is the organic compounds. So this organic compounds, first of all, it is the carbon source. So this carbon source, it is very much essential for the heterotrophic mode of nutrition. Example, it is the sucrose, fructose, glucose, maltose, galactose and sorbitol. So these are the carbon sources which are added to the medium. At the, it is added at the amount of 20 to 60 grams per liter. And vitamins are thiamine, that is vitamin B1. It is very much essential for the synthesis of amino acids, and it also acts as a coenzyme for the metabolism of carbohydrates. Then next is the myoinositol. It's also it's a sugar alcohol, but it is often grouped under the vitamins. It is responsible for the synthesis of cell wall components like pectins and phospholipids. Next is the nicotinic acid, that is vitamin B3. It also acts as a coenzyme. Also pyridoxine, vitamin B6, it also acts as a coenzyme. And next is the amino acids. First of all, it is the L-glycine. It is very much important for synthesis of purines and chlorophyll. And then L-glutamine, asparagine, serine and proline or other amino acids which are added to the medium as organic compounds. And next is the complex organic compounds. These complex organic compounds means these are the compounds which are added to the medium, but we do not know the exact composition of the nutrients which are present in the compounds, in these compounds. That's why it is called as the complex organic compounds. Example is the protein hydrolysates, that is casein hydrolysate. It is used as a nitrogen source and it contains many amino acids and organic nutrients. Then preparation from the nuts, especially it is the coconut milk. It is also a source of plant growth hormones and other organic nutrients. Then fruit juices like organic orange juice, then tomato juice, grape juice, pineapple juice, all these are used as the complex organic compounds in plant tissue culture media. And in case of inorganic nutrients, first of all, we will see about the macronutrients. So in this macronutrients, 6 elements are very important. That is nitrogen, phosphorus, potassium, calcium, magnesium and sulfur. So nitrogen is added in the available form as potassium nitrate or ammonium nitrate. So this plays a very important role in the protein synthesis. And next is the phosphorus, which is added as potassium dihydrogen phosphate, which is used for activation of nucleotide synthesis, and potassium as potassium nitrate, essential for activation of many enzymes and also for maintenance of ionic balance in the cell. Then calcium, it is added as calcium chloride. It is also a cofactor and also it is largely bound to the cell wall and cell membrane, and also it is used for maintaining the cation-anion balance. In magnesium, it is added as magnesium sulfate, essential for photosynthesis and other enzymatic reactions. Sulfur, it is added as magnesium sulfate or potassium sulfate. Its role is in protein synthesis. Next is the micronutrients. So, these micronutrients comprises of zinc, manganese, copper, molybdenum, boron, iron, then cobalt and chlorine. So, zinc, it is added as a zinc sulfate. It is a component of number of enzymes. It acts, it is responsible for protein synthesis, and also it is responsible for certain amino acids, mainly tryptophan. And then manganese, it helps in photosynthesis, that added as manganese sulfate. And then copper, as added as copper sulfate, it plays an important role in the electron transport chain at the time of photosynthesis. Then molybdenum, it is added as sodium molybdate. It participates in the conversion of nitrate to ammonium. Then boron, it is added as boric acid. It is required for the synthesis of cell wall and cell membrane. Then iron as ferrous sulfate, it is responsible for formation of proteins and also biosynthesis of chlorophyll. Then cobalt added as cobalt chloride, helpful for nitrogen fixation. Then chlorine as calcium chloride, it is added, and it is controls the osmoregulation of cell development. Then apart from that, is activated charcoal is also can also be added to the tissue culture medium. So, this activated charcoal is added at a proper concentration, that is 1 to 2.5 milligram per liter only. So, it facilitates for the efficient growth of the culture. Mainly it is responsible for removal of some of the toxic compounds, toxic or some inhibitory compounds that are produced by the tissue culture is removed by the activated charcoal, but the concentration should not exceed this level, exceed this level, because sometimes it may absorb the hormones which are added to the medium, and thereby inhibiting the growth of the cultures itself. So, we should be very careful in adding the activated charcoal. Also, this antibiotics the same way only. So, low concentration of antibiotics, that is 100 to 250 milligram per liter, either streptomycin, kanamycin or ampicillin, any antibiotic can be added based on the expected contaminants in the medium. As far as possible, addition of antibiotics should be avoided in the medium, should not add in the medium, but you can go for treatment of the explants using the antibiotics. Next is the plant growth regulators. So, this plant growth regulators, most important one is the the auxins as well as the cytokinins. First of all, we will see about the auxins. So, auxins, it is categorized into two types. One is the natural auxins as well as synthetic auxins. First of all, natural auxins, it is the indole acetic acid. It is a weak form of auxin, and it is added at high concentrations because it is very weak, 0.01 to 10 milligram per liter. And indole butyric acid is a more active form of auxin, and is added at a concentration of 0.001 to 10 milligram per liter. In synthetic auxins examples are naphthalene acetic acid, then 2,4-D, then 4-amino 3,5,6 pyridine carboxylic acid, that is picrolam. So, all these synthetic auxins are added in 0.001 to 10 milligram per liter. The role of these auxins is at low concentrations, it is responsible for initiating the root form. It is helpful for root formation at low concentration, but at a high concentration these auxins are helpful for forming the callus. Then cytokinins, these are responsible for growth promotion both in vivo and in vitro conditions. So, here also the classification is as natural cytokinins and synthetic cytokinins. Natural cytokinins are zeatin and 2-IP. 2-IP is that nothing but isopentenyl adenosine. Synthetic cytokinins are dihydrozeatin, kinetin, benzyl adenine and thidiazuron. Here the role of cytokinins is, so at a low concentration this growth is favored, but at a high concentration it induces the formation of adventitious buds and suppress the rooting. So, this picture shows you about the role of auxins as well as the cytokinins. Auxins are responsible for cell elongation, apical dominance, adventitious bud formation as well as somatic embryogenesis. Cytokinins are responsible for cell division, shoot initiation, axillary bud formation and growth. Then a low auxin and high cytokinin, it is helpful for shoot formation. An intermediate ratio of auxin and cytokinins is responsible for callus formation, and low cytokinin and high auxin is responsible for root formation. So next hormone is the gibberellins. So this was used for stem elongation in certain species. So another one is the abscisic acid. This has been used in tissue culture of certain woody plants. And finally, it is the gelling agents. So these are the agents which are helpful for solidifying the tissue culture media. Mainly the gelling agents are agar, agarose and gelangams. So they increase the porosity of the medium, and also it is helpful for the gaseous exchange of the medium. So gelangams, it is example is the gelrite as well as the phytogel. So apart from these hormones, next is the pH of the tissue culture medium. So pH of the tissue culture medium is very important to maintain. So it determines the acidity as well as the alkalinity of the solution. So the pH affects mainly it determines the viability and proper functioning of the plant cells. So this may disrupt if the pH is too high or too low. So if the pH should be appropriate, that is 5.5 to 6 is the pH in which the tissue culture tissue culture should be done. The next another one is the pH affects the solubility of various salts. So if you are altering the pH to below 5.5 or above 6, then it will inhibit the solubility of various salts. Then uptake of various nutrients are also hindered if the pH is altered. And gelling of agar also takes place at optimum pH. So these are the issues if the pH is not maintained properly. So care should be taken to maintain the exact pH of the medium. Then next is the environmental conditions. So in this environmental conditions, nutrient medium, then aseptic conditions and aeration of the culture is very important. So that is the proper nutrition should be available for the explants to grow. Then aseptic condition means the sterile condition should be maintained in the lab, as well as the medium should be very sterile. And aeration of the culture is very important. That is, it should be kept in a shaker if it is the callus culture or suspension culture. Coming to the plant growth chamber or the culture room, which is helpful for maintaining the culture conditions. So first of all, light is very important. So the culture conditions which has to be maintained in plant tissue culture is light, then the duration of that is photo period, then temperature as well as the humidity. All these are the culture conditions which has to be maintained in the tissue culture room. First of all, we will see about the light intensity or the duration cycle controller. So here it is the mostly cool white fluorescent tubes and incandescent lamps in the ratio of 3:1 is kept, and the light intensity should be 2000 to 2500 lux light intensity. So light and dark cycle, it should be maintained for 8 hours. 16 hours light and 8 hours dark should be maintained. So temperature control is done by platinum temperature sensor, and it should be maintained at 22 to 28 degree centigrade. Then humidity is also maintained by using the humidifier, and the humidity should be around 60%. So aseptic condition is very important in the case of plant tissue culture lab. So you have to use air curtain wherever there is entry points. So that should be maintained so that it will prevent the entry of outside air into the plant tissue culture lab. And also all the media should be stored in the cabinets in a separate area. And also for washing and cleaning of the equipments, everything should be done in a separate area. And also in the provided in the corridors, you can provide UV lights during the nights. So providing UV lights in the nights, it will eliminate all the microorganisms present in the culture lab. So how to develop the aseptic environment means, that is the equipments which are used for aseptic environment creation is, first of all, it is the important one is the laminar airflow chamber. So laminar airflow chamber, here what is the principle here is, it is that high efficiency particulate air filters are present. So they have the pore size of about 0.3 micrometer, and the efficiency is 99.99%. So it will prevent any entry of any fungal spores or bacteria. So that is prevented. Here in this laminar airflow chamber, the air pressure is maintained at 12 bars. So that is controlled by a manometer which is present in the laminar airflow chamber, and provided the UV light is there, and that should be switched off for about 30 minutes before starting the work. So in this laminar airflow chamber, there are two types. One is the horizontal laminar airflow chamber and vertical airflow chamber. In this horizontal one, the blower is present at the bottom. From the blower, the air is sucked inside into the laminar airflow through the pre-filter. It enters through the pre-filter. From the pre-filter, in the pre-filter, the dust particles are removed, and from the pre-filter, the air enters into the HEPA filter, that is the high efficiency particulate air filter. In the HEPA filter, it will eliminate all the microorganisms, either fungal spores or bacteria present in the air or removed, and the air which enters into the laminar airflow chamber will be the clean air. So it is also very much helpful, and this is horizontal airflow chamber, in which the air is flown from backside to the front, whereas in the vertical airflow chamber, from the top the air is flown towards the bottom. After that, that is explants. We should be concerned about the explants. So surface sterilization of explants is very important. So these are the chemicals which are used for surface sterilization of the explants to remove any contaminants present in the culture. So there are many 1% of sodium hypochlorite, 7% calcium hypochlorite, then 1% bromine water, 70% ethanol. Then mercury chloride, hydrogen peroxide 10%, 1% silver nitrate. So these are the examples of surface sterilizing agents. It's not that all the sterilizing agents should be used. Any one or two can be used for surface sterilization of the explants. Overexposure to the sterilizing agents may also help in may also cause the death of the explants. So we should be very careful in standardizing the sterilization process. This is the flow chart which has to be followed for sterilization of the explants. First of all, you collect the explants, wash in running tap water for 5 to 7 minutes, then you wash with the tween 20 for 5 minutes, then remove the excess detergents. Then to avoid the contaminants, bacterial as well as the fungal contaminants, you have to treat with streptomycin and bevacizine for 15 to 20 minutes, and then wash to remove all the disinfectants, and finally you can treat with any one of the sterilizing agents, and then you can take into the laminar airflow chamber and use it for inoculation in the tissue culture media. And there are three types of sterilization process. One is the dry heat, wet heat, as well as the filter sterilization. In dry heat, the glass was metal tools and other articles are wrapped in the aluminum foil, and it is kept in the hot air oven for about 3 hours at 140 to 160 degrees centigrade. Here in this, for dry heat sterilization, you should not use the media or any plastic wax should not be used. For wet heat sterilization, it is used for sterilizing the media, that is at 121 degrees centigrade at the pressure of 15 pounds pressure for 15 minutes. Nowadays, modern autoclaves are developed which can reach up to 132 degrees centigrade, and the pressure is 25 pounds pressure. The capacity of the quantity of the solution, the medium that has to be filled in the container is about 40% only. Next is the filter sterilization. It is otherwise called as the cold sterilization. So this is used in the cases where mainly this growth hormones are thermolabile in nature. In those cases, we have to go for filter sterilization. The porosity of the filter is around 0.2 to 0.45 micrometer. So in the case of dry heat sterilization, the equipment used is a hot air oven, 160 degrees centigrade to 180 degrees centigrade. At the roof of the autoclave, there is a hole where the thermometer is fitted, and this thermometer is helpful for regulating the temperature. There is a regulator knob in the hot air oven, which you can set the regulator so that the exact temperature can be maintained. And for heating purpose, there is an electrically operated heater which is fitted at the base of the equipment. So this is the picture showing you about the hot air oven, which has got the that is the heater at the bottom as well as the thermometer at the top, and also there are so many racks in where you can keep all the glasswares or any other materials wrapped in the aluminum foil. For wet heat sterilization, autoclave is used. Here this is an autoclave that it has got a metal body inside which there is the internal heating system, that is coil is present at the bottom, and at the top it has got the lid, which is the lid has the pressure gauge, safety valve as well as the pressure release valve. All these things are present in the lid. This can be used for sterilizing the medium. So the volume of the solution and the duration of sterilization is very important. So if you increase the volume of the medium, then actually you have to increase the duration of the sterilization process. For example, if it is 20 to 50 ml, you can the duration of sterilization may be 15 minutes at 121 degrees centigrade. Actually, this table shows you about the quantity of the solution and also the duration at which the sterilization should be carried out. For example, if it is 2000 ml, that is 2 liters, then the duration is 40 minutes. You have to increase the 40 to 40 minutes duration sterilization. Only then there will be the complete removal of or killing of the microorganisms. So this is the picture showing you about the filter sterilization unit, and this filter sterilization, I told you, it is otherwise called as the cold sterilization. For filter sterilization, there are two types of filters that are used. One is the membrane filter, another one is the glass filter. So membrane filter example is the nitrocellulose membrane with porosity of 0.22 to 0.45 micrometer. So this is helpful for removing all the microbial contaminants. Whereas in the case of glass filters, the example is the sintered glass filter, which is available in different grades G1 to G5, and G5 has got the porosity of 1 to 2 micrometer. This is also useful for bacteriological purposes. Here, as I told, it is useful for sterilizing the thermolabile solutions, which are very much heat sensitive, especially vitamins, amino acids, as well as the plant growth regulators. So this whole unit, so this shows there's a flask. There is a flask at the bottom. So this is called as a vacuum flask. This vacuum flask is connected to a suction pump. So through the suction pump, what has happened? Suction is created so that at the top there is a funnel where you can fill it with a solution which has to be filter sterilized, and because of suction force, it will be filtered and the filtrate will reach the bottom. So this can be then added to the medium which is in a semi-solid state and then used for culturing. So in the case of membrane filters, that is the pressure is applied from the top, whereas in the case of glass filters, this suction pressure is used. So this is the one which is very important. We have to take care that growth hormones are sterilized by using the filters. Then another one important one is the sterilization of tools. There are so many tools we are using in tissue culture, especially surgical blades, scalpels, then forceps, etc. So all these cannot be kept in the hotter oven. So because we should avoid keeping it in the hotter oven, because so the blades and all, that the sharpness will be reduced. So that is why you should not use the hotter oven. Mostly the preferred one is the dipping of the surgical blades or scalpels or anything in 80% ethyl alcohol. That is the ethanol. You can immerse it for about 10 to 15 minutes, and then you can show it in the flame and then use it. So that is the procedure that is followed. Apart from this, nowadays mostly in all the labs they are having the glass bead sterilizer. So glass bead sterilizer, it works under the principle of dry heat sterilization. Here this initial stabilization temperatures, you have to switch on the equipment and leave it for about 30 minutes. So by the time after 30 minutes it will reach around 250 degrees centigrade. So at this 250 degrees centigrade, just if you immerse the scalpel or this forceps just for a few seconds, it will be sterilized. All the microorganisms will be removed. So that is very much useful, and it can be simply kept in the laminar airflow bench itself, and it can be used. And most importantly, use of fumigation is, but most of the labs they follow fumigation, but that should be discouraged because it is very carcinogenic in nature, so that you have to avoid it. So it's always better you can clean all the benches using 80% alcohol. So that itself will remove all the contaminants in the work area. So if you are using fumigation, you have to displace all the bottles to somewhere, and then you go for fumigation. Fumigation, by keeping all the cultures inside is it is detrimental to the culture also. So that should be avoided. So better clean all the surfaces using alcohol. Then that is wiping of the working place with 80% alcohol is sufficient for regular use. So these are the techniques which has to be followed for sterilization of all the tools. So that's all about the culture media preparation, culture conditions, as well as the sterilization techniques. Thank you.